for the invitation. I didn't know of this initiative. I guess there's quite a few now online, but for the immune macrophage community, I just saw that you have had a very nice list of speakers. So it's an honor to join them. And I actually, it's too bad the time difference is a little bit tricky. I would have loved to join some of them because there's a lot of cool stuff discussed there. So I'm a little bit new to the heart, not new to the immune system, but new to the heart. So there's always good feedback that I hope to get from you guys. And this, of course, the spin that we're immunologists, and we just came to the heart a little bit later. We do macrophages. We have some other interests on neutrophil biology and hematopoietic stem cells, but you know, in the context of this series, I thought this would be more appropriate. This I'll tell you mostly about what was published, but hopefully with giving you a little bit of our perspective and being able to discuss with you where this is going, and then just a few slides to discuss really what we're thinking this could take us, and this it's probably where your feedback would also be very interesting, because this more than the work itself is where we look for and what possibilities this opens. So macrophages in the heart, and before I start, I just wanted to make point, of course, acknowledge the team behind this, and especially Jose Angel. I mentioned him later. He's been a student now, a postdoc in the lab for quite a long time now. He decided to spend a couple of years more in the lab, following up some of the work that I saw later. But really, this is a team effort, not only from Jose Angel, who is leading this, but you'll see there's a lot of techniques and ideas and methods that we use. One good thing of the THNIC, here is a small institute. We're 31 faculty, and that has a lot of benefits, and one of them is that it's quite easy to collaborate with people with very specific skills and knowledge. And this really is, I guess this is where research in the 21st century is going. There's so much going on and so many techniques and concepts that one has to really reach out. And in this case, it's easy to reach out because it's a small place. So I'll acknowledge later also Antonio Enriquez, who was the other half of this. It's a lot of mitochondrial biology, so he was responsible to guide us in that regard, but also collaboration with developmental biologists, clinical cardiologists like Borja and Hector, autophagy experts, proteomics experts. So this really is I think it's worth making the point that research nowadays, or at least with the resources that we have here in Spain, requires that you reach out. And I do enjoy and I have fun with collaboration, so that's a plus. So the question that Angel took, as I mentioned, he came, I don't know, I think it was like 7 years ago. It just sounds like a very long time, as a master's student, and then was a technician for a year, and then started his PhD, and now he's a postdoc. So we'll have to get married one of these days. We've known each other for so long. So Angel took up this project, and the basic question is summarized in this movie. So this is a clear heart from a CX3-01 GFP animal, and what you see in green are macrophages, and in white endothelial cells, vessels. And this is to illustrate that when you take a naive heart, so a healthy animal, and you look at the heart, what you find is an extraordinary high density of macrophages. And Matthias Narendorf published 3 years ago work on specific areas of the heart where he saw a higher density, but the density in the ventricular wall is already very high. And one question, of course, that comes up is what are macrophages, which are a canonical immune cell, doing in a healthy tissue in such high density? So that's a naive question, and now we know that this applies to many other organs and tissues. Why do we have macrophages when we gave the tag of immunity? And as you will see, and I think it's quite clear now, is that immune cells, cells that we call immune, do many more things than just defend against pathogens. And in this case also, how they contribute cardiac physiology. So that was the question of young Jose Angel. And this will be a collaboration, as I mentioned before, with Toño Enriquez and Ana Victoria, which are co-leading authors with us. Because as we came from the view perspective of an immunologist interested in macrophages, we just got into uncharted territory for us, and starting to find all sorts of things that we knew nothing about, like cardiomyocytes and metabolism, mitochondria, and autophagy. So that was a bit scary, and that was also very educational and fun. And this is what we did. So anyway, there's so many, going back to the question, there's so many macrophages that in fact we play with the idea that if you put one dot, a small dot per macrophage, you can basically reconstruct the whole cardiac tissue. This is how many of these cells are, and what are they doing there. So giving a step back, I like to make the point of this new wave of immunology that we look from the perspective of 2021. So sorry, this is a COVID number. It's 2021, and how we perceive things in a different way from what we just started studying them just like 20 years ago, when we had this very hierarchical tree, and we just devised the myeloid lineage and the lymphoid lineage, and neutrophils and neutrophil, monocytes and monocyte, dendritic cell, and so on and so forth. Things get a bit more blurry, but also more interesting with this new perspective that the choices to become one cell or the other are much more plastic and driven by environmental changes, but also that the choice to become one cell or another start early on, and might be imprinted clonally even. And when the cell matures, as is the case of monocytes or macrophages and even neutrophils, there's plasticity even when the cell has matured, and it can become something else, at least in functional terms, even if it's still tagged with the same name. So plasticity is a key feature of immunology and immune cells as we see them now. And this is very particularly true in the case of macrophages. And we, not we, but the field has been developing quite strongly over the last 20 years or so, I would say, with the realization of this M1, M2 dichotomy, and then the realization that both the origin and the functional specialization of macrophages in different tissues really is very different. So again, the same name tag of macrophage, we have very different cells, Kupffer cells that might be aiding more hepatic metabolism in the lungs, alveolar macrophages, which remove surfactant proteins, those in the bone that help maintain recycling of the bone in the form of osteoclast microglia, and so on and so forth. So it was very clear already from a number of years ago that there's a high degree of specialization. And one macrophage subset that came late a little bit to this crazy race of describing immune cells in tissues were macrophages from the heart. And these were originally described by the group of Nadia Rosenthal. You know, we all suspected that they might be there, as he first put attention to these cells in 2012, so not that many years ago. Just by showing that, you know, there was a dense network, not making any claim on what they were doing, but rather showing that there was a specific profiles for these cells, this M2 kind of profile. And it was a few years later that, as I mentioned, Matthias Narendorf made the point that in a steady state, those cells have functions in aiding the electrical conduction of certain networks of cardiomyocytes. And all the rest that was done with these cells was basically in the context of injury and inflammation, so myocardial infarction, for example. But not, you know, still the question was, why do you have so many of these cells in these cardiac walls in the steady state? You know, this is not the atrioventricular node that Matthias described, and this is not an inflamed tissue. So what are they doing there? And this is what drove our work. So one thing that Jose Angel did was to use imaging, which we love in the lab. He created these mosaic mice. Oh, I'm sorry. secreted these mosaic mice, where only a few cardiomyocytes will become fluorescent, and this is very nice because you can see nicely the architecture and the structure of the cells, and in this case, macrophages, C68 positive macrophages around them, to show that there's a clear network or high density of macrophages surrounding cardiomyocytes. And this is an example of one of these that allows to actually quantify the ratio of macrophage or cardiomyocyte to macrophage, and making the point, which is showed in this image, that there's quite a few macrophages, and an average of 5 around each individual cardiomyocyte. And then, because macrophages extend a lot of protrusions, they also reach out to a number of different cardiomyocytes. So really, quite a thick network of immune cells around cardiomyocytes. And this is, of course, one of the points that drove our interest in understanding the function of these cells. So when Jose Angel came as a young and naive master student to the lab, I proposed him a simple experiment, which was, hey, we just got these C169 DTR mice, and they're very cool and efficient at depleting macrophages, not only in the heart, but in other places. We've seen that they deplete very well. Can you just check if they allow depletion of cardiac macrophages? Because if they do, then we have a model to start studying the question of what is their role. So he did this kinetic analysis and very nicely showed that the model works very efficiently. So a single injection, we deplete them very efficiently, 80 to 90% of them. And if you sustain depletion, in this particular case, you can successfully keep the depletion up to for 21 days. After that, some of the mice start dying. So we thought this would be 3 weeks would be good enough to see the effect of long-term depletion. And that was his first experiment in the lab and for this project, which he published a number of years later. But it's interesting to see how things start. And I discuss with him often how things start with one question and one naive approach, and then they start getting complicated and interesting. But he never complained about it, so I'll take it as a good sign that he liked the process. The science takes a long time. So now that he had the model, again, one of the first experiments that he ran is, since we're in a cardiovascular institute and there's a lot of tools to measure cardiac function, not that we are experts, but the tools are there and the people with expertise are there. We just run a relatively simple experiment where we depleted macrophages for 3 weeks with diphtheria toxin, or control mice with DT, which shouldn't do anything, just to control for the toxin, and just measure cardiac function or contractile and relaxation cycles by using echocardiography. So you will see this in the slower motion, of course, than real life, and you will see what a healthy heart looks like. So this is, for us, was aligning for the first time that I saw this. For cardiologists, this is an everyday thing, but for me, learning of how the wall contracts and relaxes, and what this looks like in normal conditions. And this is what, in this case, looked like 3 weeks, the same mouse 3 weeks later. And you can see that there's some obvious changes here. Some affect to the size of the left ventricle, which is quite clear. I think you can see this quite clearly. Seems to be a bit contractile, shrunk. And then both the contraction cycle and the relaxation cycle are clearly compromised when you look at this. And you can quantitate this and give numbers to show that really there is a progressive decrease. This is doing measurements every week. A progressive decrease in left ventricle volume, a decrease in the EA wave ratio. So this is a measure of relaxation of the left ventricle, so diastole, with also a gradual and significant strong loss of diastolic function, without, in principle, apparent effect on the ejection fraction, so on systole. So we decided, and this was an advice of one of the reviewers, to look this in more detail. So we used thermodynamic analysis, and we inserted the probe so we could measure specifically and with more resolution changes in pressure in the chamber. And this basically told us that indeed the diastolic function was compromised, but also the systolic function. The systolic function was compromised when we look at these temporal changes in pressure. This is very clear, and the overall outcome was that the cardiac output was also strongly decreased, making the very simple point in our immunologist heads that if you deplete macrophages from the heart, there's a progressive loss, and really is progressive, loss of cardiac function. So that was the observation, and that's cool. Of course, now we only need to figure out why. So that was the big challenge, the second challenge that Jose Angel found. We were a bit stuck. Of course, we were not experts in cardiac physiology, but we resourced to something that we do often in the lab. Is we don't know what's going on. Let's take a look at the protein level rather than the transcriptome level. This is in part because we have a very good group in unit in proteomic analysis, but also because in our experience and now more in the heart, changes in protein are much more informative in many cases of the actual functions rather than gene expression. So same thing, we just did depletion for 3 different time points, and then we taken 21 versus 0. We just look at what pathways, functional pathways were affected. And something that became very clear early on was that those pathways involving mitochondria were strongly affected. So there's 4 pathways marking yellow here that I'm showing related to general mitochondrial dysfunction, Autophagy activity, TCA cycle, FA oxidation. So very clear heart-specific sort of processes mediated by the heart, which is most prominently an oxidative in terms of metabolism, oxidative organ. So there seemed to be from this proteomic analysis a link between macrophages and mitochondria, because when we depleted, these proteins became prominently changed. So the second thing that we were quite fan of in the lab is to use imaging. Seeing is believing, and we wanted to see what the hearts look like, if we could see any ultrastructural changes when we did these depletions, and there was such strong changes in proteomics and in function. So this is transmission electron microscopy images that Angel took, comparing a healthy heart and one that had been depleted of macrophages for 3 weeks. And you can already see that these electrodense structures here, which are mitochondria, are very, very distinct in cardiomyocytes, round and big and electrodense, change quite a bit after depletion. So after 3 weeks, they become less electrodense. Instead of having more of these lines or isolated mitochondria, then you get these branches of mitochondria. And if you look a bit closer, you also see that they change not only in density, but also in size and shape. So you can record all these, and speaking with the mitochondrial experts, they tell us that these are actually measures typical changes that you see in dysfunctional mitochondria. So on one side, we saw this increase in the number of individual mitochondria. We could confirm with staining with TOM20, which is a marker of the outer membrane of mitochondria. By measuring citrate synthase activity per milligram of protein, you see an increase, moderate but significant increase in the heart, but not in the liver. And this control was good because the DTR model that we use depletes macrophages not only in the heart, as I mentioned before, but also in the liver and the spleen. So the system is not absolutely clean. However, this change in mitochondrial contact, or in CS activity anyway, was specific for the heart, but not for the liver. And then by looking the structure of mitochondria, we saw that there was an increase in area and a reduction, strong reduction in crystal area, which are very prominent indicators that something is wrong with these mitochondria. So, okay, now we know that there's some connection between macrophages, cardiac function, and possibly this might be related to alterations in mitochondrial quality in these animals where we deplete macrophages. So what we did next is, well, they look abnormal. Let's measure the primary mitochondrial activity. And what we did is isolate mitochondria from control of macrophage depleted hearts and measure ATP synthesis using different substrates. And that didn't matter which substrate we used. We always got a reduction in the yield of ATP when you deplete macrophages. So again, highlighting that there's something wrong. These mitochondria are not normal, they're not functioning well. And this also suggested that then if mitochondria are not optimal in these mice and they're not able to do what they are supposed to do, which is produce ATP primarily from the use of fatty acids, then maybe the demand for energy will require the hearts to start using an alternate source of energy. And typically you look for glucose uptake, which there's some, of course, in the steady state heart, but in conditions of stress, for example, under different models of mitochondrial dysfunction, they would use more glucose and you should be able to see more of this uptake. This is another typical measure of metabolic dysfunction in the hearts. So this is what we did here. And it was confirming to see that compared with the control group, when you depleted again macrophages for 3 weeks, the heart started taking much more glucose. So again, suggesting that there was a switch in the type of metabolite that they use as the primary source of energy. So overall, mitochondria are not looking good. This might be causing a cardiac dysfunction if you don't have macrophages. But of course, we still don't know what's going on, what's the connection between cells outside of the cardiomyocyte and mitochondria that are inside. So very naively, again, we asked, what is it that macrophages might be doing? And one thing they primarily do is fight to phagocyte those material or cells from the environment. So we asked if they were phagocytosing in the heart, like they do in other, for example, lymphoid organs where they're constantly taking apoptotic cells. So if they would be phagocytic there or just doing something else. And if so, would be their primary, their favorite source of target cells of food. So this is what this experiment was about. So what we did is to establish a system in which we had non-fluorescent macrophages and all of them to eat different things. In the first experiment, using bone marrow transplantation, we have everything is fluorescent except macrophages. And then if we take them, we see that they are actually quite phagocytic. So around 60% of them are actively have phagocytosis. This is specific because monocytes do not take up material to the same extent. And this is just a classification of different subsets of macrophages based on specific markers. But this is not important. Just look at the red part and you see that they're quite phagocytic in the myocardium. So that was interesting because not necessarily something that we expected. There should be little cell death in the heart. And the second question is, what is it that they're eating in the heart? And for this, we generated different tricks to measure, to label different target cells. We use parabiosis, so only circulating cells are fluorescent. Then we see some degree of phagocytosis, not a lot, but some degree of phagocytosis. If we label endothelial cells with tomatofluorescent protein, then we see low-grade phagocytosis, at least overall. There's some for this subset, but overall, they don't like to eat much of endothelial cells. Now, the surprise was that if you label cardiomyocytes, which are among the longest lived cells in the organism, so these cells pretty much shouldn't die, then you get a lot of fluorescent signal in macrophages. And it was striking because, you know, there's a whole blast of literature of how long, how often cardiomyocytes replace. And the answer is that they replace almost none to a very, very low extent. So for all purposes, you're born with the same number of cardiomyocytes that you die with. And this is interesting because if they don't die, then what is it that macrophages are taking up? You know, where is this material coming from? Are they eating the cells alive? That doesn't make a lot of sense. But this was the observation that the preferred food was derived from cardiomyocytes. So how does this work? And then we went again into imaging. So Angel did simple confocal microscopy, and what he found, and this was interesting, was that the signal, the reflorescent signal that was specific for cardiomyocytes, because we use specific driver, was in the cells in cardiomyocytes, expected, not in macrophages, but it was also present in these vesicles that are floating around here. And in fact, macrophages tend to associate to this, to the presence of these X structures. And this sort of like the joke with Jose Angel was that that he needed to chase the crumbs, you know, a little bit like that tail where the hint is the crumbs and you can get to the place that you're looking for. In this case, really was these vesicles that were there that look like just debris, something that you don't look at. They were the main hint, because what we thought is they shouldn't be eating these cells alive. That doesn't make sense. But what are these particles that they're eating that seem to be derived from cardiomyocytes? So we christened these vesicles with a name inspired on the work of Monica Driscoll. We call them Exophys. So Monica described these structures in C. elegans a number of years ago as vesicles that were produced by a specific type of neurons. And she proposed that these were these exophys were specialized in eliminating aggregates, protein aggregates, so in general material that the cell does not want. So that was great to put this name. It was a bit serendipitous, but it gave it was important for us because it gave a little bit of a purpose. We didn't know what they did, but we had this inspired by this, that there might be a way to get rid of material. And this is just an image illustrating a macrophage taking up one of these exophys. So we did see many events of this phagocytic uptake. And this, of course, was important because this connected with the elevated phagocytic activity that we saw when we labeled cardiomyocytes in red. So we thought that this was the preferred food of macrophages in the myocardium. Now, this idea that macrophages eat material from cells that are otherwise alive and healthy is not new. And Greg Blenke is there in California, actually, in La Jolla. Is an expert in this. I was also a collaborator in the paper, and he's proposed that one role for phagocytic cells, not only macrophages but also some types of epithelial cells, is to take up material from surrounding parenchymal cells, for example, a neuron or a retinal cell, and this is a way in which these cells get rid of material that they don't want. For example, all photoreceptors in the case of the retina. And it's very interesting if you disrupt this process in any way by limiting the epithelial cells or the receptor that mediates this uptake, then the animals go blind. So this is relevant to keep the parenchymal cells happy. And this was a little bit the idea that we were playing with. So this process has been described, and they're eating this material, and what is it in this material that they're eating that allows us to learn more about what's going on. So went back to electron microscopy to try to find these structures and see what was inside of them, and this is what we found. These structures similar in size and in shape to what we had seen by confocal microscopy. And now, if you zoom in, you can see quite clearly that there's some sarcomeric fragments, so really they seem to be derived from cardiomyocytes, but mostly these round structures that are none other than mitochondria. We know they're mitochondria because in many cases you can still see traces of crystin because of the shape, size, so it was an easy guess. And something similar, and this was the only thing we did with humans, that was a fun experiment and important, I think, something similar with these sort of vesicular bags containing remnants of mitochondria in human myocardium. So this appeared in otherwise healthy hearts, at least in the case of the animals. This, of course, was from older patients undergoing valve replacement. But in this case was healthy hearts, and they were containing these bags. So this already gave, this is very interesting because it gave unexpectedly a connection between what we saw with the proteomics at the beginning and with the observation that mitochondria were different. And now it turns out that these bags contain mitochondria. So we start connecting a little bit the dots between the different observations. So we wanted to know more about this. Of course, those images, they're nice, and I think they're quite enlightening that really this exophys might be a way to transfer materials such as mitochondria. But I think a breakthrough in this project came from when Angel became able to purify exophys from the myocardium, from means myocardia, using just a trick of differential centrifugation system. This has been published recently in this STAR Protocols new journal, if you want to take a look at that. But basically, the fact that he was able to purify with quite a good accuracy Exophys was great, because now we could start asking in a more quantitative way, what is it that these Exophys contain. So we ran proteomics on this material, and what we found was quite striking and confirming. On one hand, that mitochondria was the preferred content. So here what we're doing is compare the proteome of Exophys versus the total cardiac tissue. So we have a reference, okay? You know, it's the content has to be relative, I think, compared to where it comes from. So in the total cardiac tissue, you see already a lot of sarcomere suspected and a lot of mitochondria. So this is what these cells have mostly inside of them. But now when you look at Exophys, what you see is that basically all other compartments, proteins from all other compartments are reduced, except for mitochondria, which become massively enriched. So this is mostly what these structures, what these vesicles are containing. And this is just a validation of some of the proteins that we found here. So that's one thing that we found, that, you know, confirming the imaging data. Proteomics told us the same thing. But also there were, if you look at this specific graph here, there's some, you know, in blue, all these mitochondrial proteins are enriched in Exophys, except a few of them. And of course, you know, a naive, ignorant immunologists, we didn't know what this meant, but Tonio, you know, our mitochondria guru and collaborator with this, told us it does all these proteins that were specifically reduced in Exophys, of the many mitochondrial proteins, were proteins involved in quality control of mitochondria. So if they're lost from mitochondria, it's a sign that those mitochondria are typically dysfunctional. So your typical canonical molecule would be cytochrome C, which is right here. So the same thing applies to this bunch of mitochondria. So we learned not only that mitochondria were preferentially enriched in these structures, but also that the mitochondria that were there might be dysfunctional. So now we continue connecting the dots, that this might be a way, like Monica Driscoll described in C. elegans, to eliminate dysfunctional unwanted material. In this case, would be dysfunctional mitochondria. That was what the data suggested, but not demonstrated. So how did we try to address this point? Are these mitochondria specifically dysfunctional, and this is why they end up in this vesicle spell-out of the cell? So we use an image criteria, just looking for features of abnormality of mitochondria versus normal. And what we did, with Jose Angel did, is to then take a lot of cardiac sections and map in the cardiomyocyte, and, you know, in this scheme of cardiomyocyte, map the quality of mitochondria in different regions. The idea being that if we saw mitochondria in Exophys, that there may be a gradient of dysfunctional mitochondria or normal mitochondria from the center into the periphery. So eventually, when they come into the Exophys, they're mostly in a dysfunctional or aberrant state. So this is the work of you know, a lot of work summarized in this slide, showing that as you move from the perinuclear area into the border zone or the Exophys, you get a gradient and an progressive increase in what we call abnormal mitochondria. And of course, the opposite is true for the normal. And with this suggested again is that there's a gradient, and perhaps indicating that this is a passive, relatively passive process, then it makes sense that Exophys end up containing this higher cargo of apparently dysfunctional mitochondria. But to make the point in a more quantitative and rigorous way, what we did is then measure functionally how intact was the membrane of this mitochondria, which is needed in the end to generate ATP, using a probe that allows us to look for the polarization, you know, for the membrane potential of the mitochondria. We use fibroblasts and leukocytes as controls, but now, and you can see the response to hyperpolarizing of the polarizing agents that, like, you should see normally. But now if you take Exophys and you look for mitochondria that are specifically in Exophys, that you find is that they're unable to respond to polarization or depolarization. What it suggests is that these mitochondria are broken or dysfunctional. They just not properly functioning. And, you know, another measure is to look for cytosynthase activity in mitochondria. In all evidences, both morphologically and functional, what we found is that these mitochondria inside these exophys were dysfunctional. You know, that it gave the purpose that the cardiomyocyte was getting rid of material that it didn't need anymore. So that's, of course, that's showing that exophys contain this, and the cardiomyocytes presumably get rid of them. But you know, we needed to still to connect the dots that they were produced by cardiomyocytes and that they reached macrophages, which is what we propose in our phagocytosis experiments, and it's illustrated here. So what we did is to prove this in a more unequivocal way is to use genetic labeling. We infected animals with adenovirus, you know, with tropism for cardiomyocytes and with a specific promoter that allowed expression of this fluorescent protein specifically in cardiomyocytes, and we did the controls and so on and so forth, because this was the key point in this experiment. And we introduced KEMA, this KEMA fluorescent protein tagged with mitochondrial peptide. So we would be able to label exclusively cardiomyocyte mitochondria, and then over time see if we saw a transfer of these mitochondria into macrophages. So this was the point of this experiment, to establish an origin and a destination for these phenomena. So this is what the heart looks like. The red signal here is this mitochondrial KEMA protein, and this is a demonstration that if we wait long enough, then we see this KEMA protein in structures that resemble the exophys that I described before. And ultimately, they appear concentrated in macrophages. So this mitochondrial KEMA, which looks like these dots in a cardiomyocyte, then becomes this condensed group of KEMA proteins, suggesting that this macrophage was taking up all this material. And this is a 3D reconstruction to make this point, that the KEMA protein that is punctuating individual mitochondria here ends up here as clumps inside the macrophage. And an experiment that Angel wanted to do very specifically, when initially I wasn't very enthusiastic, is answer the question of what is the macrophage now doing with these mitochondria? Is it using it, recycling them, as has been described for its own use? You know, is this a transfer of functional mitochondria to use? We didn't think so, because we saw them to be dysfunctional. Or is this just the end of the mitochondria? This is a degradative system to eliminate dysfunctional mitochondria. This is what we propose, and what Angel demonstrated here by taking macrophages, putting them in culture, and look for the decay time to eliminate KEMA. So after a couple of days, there was no trace of KEMA protein anymore. So is this a cardiac specific phenomenon? And of course, this illuminates some of the work of the future for what we're trying to do now. And the question is relevant, again, because cardiomyocytes are very specific in terms of metabolism, inability to recycle or to enter cell cycle. They're not stem cells that we know of in the heart, or I mean, cardiomyocyte stem cells. So they have a lot of constraints that are very specific to the heart, and it's striking that this has not been described elsewhere. So we wanted to know if really the specific or the uniqueness of cardiomyocytes drove this peculiar type of transfer, or this is more of a disseminated response that we didn't know about. What we did here is together with the mitochondrial biologists, our colleagues here, is to establish this interesting system that they have. What we did is bone marrow transplantation of mice that are histocompatible. They're both B6, with one peculiarity, is that the mitochondrial DNA has a base change in one of the mitochondrial genes. So this is very cool, because now you can digest. You generate an enzyme, a resistant enzyme site that allows you to cleave that gene, and you can discriminate the mitochondrial DNA derived from the C57 animal or an NCB animal quite clearly in these DNA gels, right? So the point here is now, because the donor is NCB and the recipient is C57, if there's an exchange of protein or of mitochondria, we would be able to see exchange of mitochondrial DNA. And also, this is very cool, because when we do bone marrow transplantation, then we can take macrophages from a lot of places, not only from the heart. So this is why this experiment was useful. So this is the setup. So Angel sorted the cells and then ran this NCB versus C57 discrimination. And what we found doing this soft test is that the signal is I'm not going to say unique and specific only to the heart, because we found a little bit of signal in the skeletal muscle, but it really is quite distinctive and is strong in the heart. And for many tissues like the liver, spleen, bone marrow, intestine, there's basically no signal. We've extended this is not published, we've extended this to 18 organs now, and we're extending it more using this same approach, basically because we want to know which organs might be resorting to this sort of exchange. So this made the point that this is quite specific to the heart, to some extent to skeletal muscle, and this is something that we're working on now. So we can discuss later if you're interested in this. So this again led us to the next question: is how is this working from a cellular or molecular biology point of view? And the reason we wanted to address this, even if we're not a molecular biology lab, was that the fact that they are exchanging dysfunctional mitochondria very much is reminiscent of autophagy, right? Which is the system where that cells use to cell intrinsically eliminate dysfunctional or organelles or protein aggregates. It serves for sort of functions, but it's very typical for degradation of aberrant organelles, which is precisely what we were seeing here, except instead of seeing autophagy, what we're seeing is surrogated autophagy. So the cells don't do it themselves, they give to another cell to generate this autophagosome, or phagocytosis in the case of the macrophage. So this was our model that this might be at play here, and this is what we tested in two ways. We either gave rapamycin to block mTOR and stimulate autophagy, or we generated a genetic mouse model where we eliminated ATG specifically in cardiomyocytes to see if either promotion or inhibition would affect the production of these exophys. Another hint that came supporting the notion that this might be autophagy is that when we look, we got access to mice with a GFP label IC3 protein, which is, it's this lipidation process here where the autophagosome gets labeled with this protein. And now, because we have GFP, we can track the formation of these autophagosomes or autophagosome-like structures. And as you can see here, these exopher-like structures here are very strongly labeled. Not only this, basically all that we found here are very strongly labeled with LC3 GFP, and this is imaging, and this is the same thing by flow cytometry. So okay, this was the evidence. And now the actual experiment. So when we stimulated autophagy with rapamycin, not very specific, but still in that pathway, what we found is a significant and relatively strong increase in the formation of exophys. Not only more exophys, but also more transfer of the gamma protein into adjacent or neighboring macrophages. So this was consistent with the idea that if you stimulate autophagy and autophagy is involved, you should see this, and this is what we found. And what about the opposite experiment where we eliminate ATG7 from cardiomyocytes? And what we found here is all the opposite. Look how clean the myocardial tissue becomes. So a very strong reduction in the number of exophys and a reduction in the amount of gamma protein that is transferred from the cardiomyocyte into the neighboring macrophages. So what we concluded here is that autophagy, a particular type of autophagy at least, drives mitochondrial ejection in exophys in our model. So since this is published, and I wanted to discuss more of these initial points, I'm not going to explain everything that we did, which you can find in the paper, but go to a couple of key points. One is whether this system, that was, it seems to be specialized in recycling and elimination of material, would be responsive to a stress, which is what you would expect from a true biological beneficial phenomenon, is to be responsive to a stress. For example, as in the case of autophagy, inflammatory formation, basically all of these phenomena are responsive to stress. That's what they do. So we thought let's give a stress, a mitochondrial specific stress to the heart, so that we get more mitochondrial damage, and see if the system responds by generating more exophys. So this is what the experiment was about. We gave isoproterenol, so the oxidative stress that is induced in mitochondria ends up damaging the cells, and the mitochondria, and this is the quantification that we did to show that there was an increase in damaged mitochondria throughout the whole cell. Of course, exophys already contain damaged material, but you see that is the cell that is starting to produce to present abnormal mitochondria everywhere in its body. So now that we have more of these exophys in the cell, do we have more exophys? Is the process somehow accelerated? And this is what we found here by imaging or by quantification, that the amount, the number of exophys actually double in this setting. So yes, the system is responsive to stress, and we think that this is an important in terms of thinking about cardiac pathophysiology is an important observation. And the last question that I will present here of this particular paper is what is now thinking as an immunologist. We have the macrophage taking up this material. How does this work? What is the receptor? We have an interest in phagocytosis in the lab too, which is the mechanism of the receptor, the pathway that is mediating the uptake. And this, of course, generates knowledge, but also is not so naive, because now it also allows us to think also whether dysfunction or ways in which the immune system can affect this whole recycling system. So because I showed you before the work of Graham Lemke, who demonstrated that this particular receptor and its upstream regulator are important for the uptake for phagocytosis or recognition of apoptotic cells, or even in the case of the retina or in other settings, it's important for the recognition of these cellular structures through recognition of phosphatidylserine. So we thought that this might be at work there too. And I'm not showing here, but one thing that we found is that these exophys present phosphatidylserine in the membrane around them. So this was another hint that MERTK might be involved. So I'll just go to the experiment that worked. And what we found is that both MERTK or the LXRs that regulate its expression, a canonical phagocytic pathway also, were presented a dramatic reduction in the amount of mitochondrial KMI protein that was transferred from the cardiomyocyte into the macrophage, suggesting that this process is at least partially responsible for this uptake, and that this process is specific and the molecular terms and so on and so forth. So it makes several points, but also it allows us to see. What we predicted is now if we don't eliminate exophys from the media and mitochondria, either because we don't have the receptors or because we deplete macrophages, then what happens to the mitochondria? And what happens, and this is based on imaging, on electron microscopy imaging, is that the mitochondria start accumulating in the extracellular milieu, and this of course generates problems that I'm not discussing here for the sake of time. But one of the problems that it generates is that these structures are of course chromogenic. They're formulated. The mitochondrial DNA is recognized by the inflammatory machinery. So it really is an inflammogenic material that you don't want around. So if this material now starts accumulating in the space, what we found is the activation of the inflammasome, and inflammasome in turn has consequences in the health of the cardiomyocytes. So anyway, this is what we found both in the MERTK and the LXR and the macrophage depleted animals, and this was important to connect the dots to, but also that the animals deficient in MERTK, and I'm not showing this here, but we find the same thing, the LXR mutant mice, they manifest the same similar changes as saw before in terms of size and crystal density and quality of mitochondria in cardiomyocytes, as we saw with the macrophage depletion model, and same thing with the parallelism with the mitochondrial dysfunction. So the MERTK mutant mice compared to the controls here start taking up more glucose, we think, as a way to compensate this loss of quality in mitochondria and oxidative power. So this is the model that we presented, that we used to summarize this. The idea is that this non-canonical autophagy view. Well, let's put it this way, even if the autophagy people will probably kill me, but anyway, I'll just say, what we think in the cardiomyocyte, there's a very particular type of cell that can use clearly autophagy or mitophagy to get rid of its material. So there's a whole body of field of people interested in studying this. So I won't argue with the importance of that. But there's really a significant amount of the material that probably the cell cannot cope with, specifically in conditions of stress, but also in the state that about 60% of the mitochondria are released through this pathway out of the cell in this exophase. This phosphotylserine decoration allows recognition by macrophages through MERTK and silent elimination. If this process fails, then it's what I discussed before. Then you sort of get all sort of problems, probably many of them originating from the accumulation of free mitochondrial DNA in the milieu, which induces nasty inflammation in the tissue. So as in the last few minutes, I just wanted to discuss where do we go from here? You know, this is the typical question for a student. Now that I've published this, what do I do? And there's many more things to do. And part of the reason why Angel decided to stay a bit longer in the lab is that what is left now, what we envision is even more interesting, I think, to this initial description of the process in cardiomyocyte. So one question is, for example, what other cells or tissues? I mentioned this before. Use this sort of surrogated autophagy, where they send the material into neighboring macrophages. So I saw the example of the heart, of course, and low-grade exchange also in the muscle, in the skeletal muscle. And this is work that we're carrying out also with very interesting observations, but this is not published yet. Is another question is whether this function, support function, is impaired with aging, and this is relevant because, as you well know, this inflammagene, these alterations in the immune system and with the inflammation system, we think might be in part related with a dysfunction of the system, where if you don't have a healthy pool of myeloid cells in tissues, not only you're perhaps less immune competent, or you might be more prone to inflammatory disease, but also now you disrupt the health of the parenchymal cells, for example, in the heart. And of course, looking further on is whether this system is specific or can be adopted by pathological cells, for example, in the context of cancer. So there's several ideas from this beyond those that I mentioned, and one that we're interested in that Maria Sanchez is doing in the lab is to now ask whether this immune parenchymal relationship in the heart is established early on in the animal as the heart matures. And this is particularly interesting because most of you will know in the field that the heart is a changing organ, especially in neonatal life. So it basically, the animal is born with an immature proliferative healing heart, but as soon as 1 or 2 weeks after birth, this is completely lost. The proliferative capacity is lost, and the metabolic properties of the chain somatically change to create what we now see as an primarily oxidative organ that is unable to regenerate. So this transition is very interesting, and we're asking whether this crosstalk that was not appreciated before is important in the maturation of cardiomyocytes in these early stages of life. And this is how Maria represents the changes that you can see over time in mitochondrial quality, in cardiomyocyte quality, basically in everything that you can think of. So really, this is a changing organ, and we wanted to know what macrophages do about this. So we're still scratching here the surface, but I just wanted to share some of the things that we found. One is that Exophys, or structures similar to Exophys, both by electron microscopy, but also by quantification in confocal microscopy, which makes it a little bit easier to visualize, are present already from P1. And the changes, it's not quantitative here, but the changes in how many of these Exophys you find at different times during this maturation first month of life. But this is the structures, the vesicles are there, and they seem to contain mitochondria in basically at all stages. See, pardon? Sorry. So they contain mitochondria at all stages. So these systems is to be at work even early on when the mitochondria are not still fully oxidative and then the cardiomyocytes do not have the restrictions that they have in the adult. We did, and we've done is also a proteomic analysis, and we use this proteomic analysis to represent what is known. There's a gradual change in the properties of the heart that start accumulating particularly around day 14, postnatal day 14, where they do a switch. Don't see what the classes are here. I apologize for this. But what we see is that the big change in respiratory, in the type of respiration and loss of proliferation and increased use of fatty acids occurs around day 14. So this established this transition period as a very particular period that we want to study, towards the macrophages, and this change becomes prominent in allowing the metabolic maturation of the heart. What Maria has done is basically map out throughout the full first month of life. So this is a very time-consuming. So this really has done an excellent job, and this is, as I mentioned, this is still ongoing to show that the type of macrophages in the heart changes also, and the changes start occurring interestingly around day 14. So now what we question, this is how we classify macrophages, either with the neonatal type of phenotype, which is quite decently different from the adult type. So this is basically a replacement or a transition of the cells. That's something that we're studying. But there's this exchange in the type of macrophage. And we want to understand if it's the macrophage that will now guide the maturation of cardiomyocytes, or is the cardiomyocyte, as it changes the metabolism, that it conditions the type of immune repertoire that you have in the tissue, or both, that are taking place. So this, is a paradigm of how immune systems regulate key physiological processes. So it's just so you can see the transition using this type of approach, a transition in the type of macrophages over time. And this is one question. Another question that Angel is working on is what happens in the other side of the spectrum with age. And with age is, again, another interesting point, because the heart basically tends to go to where it started, with this glycolytic, non-proliferative, of course, but glycolytic. Metabolically, it goes back to the use of glycolysis rather than primarily fatty acid oxidation. So what Angel is trying to understand is whether this system of uptake of material starts failing with age. And so he's establishing different time points where he looks at exchange of material, as I showed you before. I'm not showing this here, but I'll tell you, he does not see any change in the uptake of material coming from cardiomyocytes using this flow cytometry trick. So it looks like the phagocytic uptake, macrophages are just fine. What he finds is what seems to be failing is not the function of the macrophages, but the number of macrophages. So here we go. Numbers matter, size matters, if you will. The number of macrophages quite dramatically falls progressively to these very low numbers if in a 2-year-old animal over time. And this is, we think, important because it shifts the importance of understanding the system and the health of the heart in how the tissue is able to preserve a healthy number of macrophages. So it changes a little bit the question. I'm aware of that, but again, we're immunologists, physiologists. We just follow the question, and the idea here is, and what we see looking evidence for, is what changes in the heart that makes it less friendly for macrophages. Because if you have less macrophages, it may be that this is the reason why hearts start failing with age. Not because the cardiomyocytes cannot do their thing, is that the support cells start leaving the place. And this is what he's studying on. Just to illustrate that the changes with age are very similar to the changes that we see when we deplete macrophages. And that's why the connection between the two become particularly intriguing. So if you remember from before, the typical aspect of a young myocardium or cardiomyocyte, and these clusters of mitochondria that appear, and when the macrophages also appear in a 1-year-old mouse, so not really a very old mouse, but in a 1-year-old mouse, there's already this change, which you can quantify here again as a loss of mitochondrial morphology and size, crest area, and so on and so forth. So I'll stop here to allow a little bit time for discussion. Again, I just wanted to emphasize the fabulous work of Jose Angel. I'm super lucky to have him working in the lab for so long. Maria is taking up, with a lot of power and energy, the work on the neonatal stage, and Albert and now Laura, who are working in different aspects, for example, other tissues, and I mentioned the skeletal muscle, that might be using a similar system to maintain mitochondrial quality and tissue health. So I'll stop here and I'll take questions. Thanks so much.
Thank you, Andres, for a fabulous tour, the force into the heart and to the macrophages. This was really amazing. We've known that macrophages are there, as you pointed out, since Nadia Rosenthal's work, but it was quite unexpected to see that they play a role in almost rejuvenating the heart, right? It's quite an interesting mechanism. If anybody has questions, we usually raise hands and then we go by the order. So I'll start with Gökhan.
Thank you, Andres. This was very nice, very interesting, many things to think about. And so, of course, I had a million questions after listening your talk, but I'll just ask just a few. One is, do you think this is part of a mitochondrial rejuvenation or disposal of damaged or superfluous mitochondria under stress conditions? And my second question is, when you're talking about this, I couldn't help but think about adipose tissue, which actually not only features many of these interactions, but also communicates with inflammatory and damage inputs with the heart. And have you considered, for example, brown and white adipose tissue in the context of obesity, whether they feature such structures?
Yeah. Well, we consider everything. We cannot do everything, but we consider everything, and so we're trying to cherry pick what we think is more relevant. So yeah, I think regarding the first question, if you think about it, and that's I guess a bit the point. You never know evolutionarily what drives this, but I guess our guess is that the heart, the cardiomyocytes are by default stressed. So mitochondria from the heart, they need to be producing ATP all the time, 24/7, no rest, no mercy, for, in the case of humans, for 60, 70, 80, 90 years. So they really, they need support for this. And if you think about it, generating ATP and in conditions of, there's always stress, I guess. It's impossible to not have cardiac stress as part of life, I imagine. This damage eventually, through ROS production, through whatever mechanism that damages organelles, that disposal or generation of these functional organelles, you can keep the cell as happy as you want, but eventually, the cell needs to use oxygen and produce RS for signaling, and you end up damaging structures in the organelles. I'm not an expert in this, but generating this functional material in any cell, more so in cardiomyocytes, which have, mechanical stress, mitochondrial stress, metabolic stress, it's probably accelerated. If you induce an additional stress, then things get even worse. So so we see this as a normal physiology of the cell. So it's not superfluous. This is how the system runs. You in this particular organ, and perhaps it's more prominent here because you have this turnover that is massive, and people have estimated this. I think the average life of cardiomyocyte mitochondria is about 2 weeks. It gives you an idea, and it's cardiomyocyte has thousands of these guys. It's 40% of the volume of this very big cell is the mitochondria. There's quite active mitogenesis. So all this tells you that the turnover is part of the system, and if you generate, you need to get rid of. So it's just that in simplistic terms, autophagy took care of everything. Canonical autophagy. Well, we think it doesn't take care of everything, at least in these cells, and this is a little bit the point that we're trying to make. Thinking about other organs, so I mentioned the skeletal muscle, and of course the fat is high up in the list. And we already have some experiments specifically looking at mitochondrial DNA transfer. As the screening that I mentioned that we've extended, looking at, among others, to white and fat, brown adipose tissue. And sure, brown adipose tissue lights up in this transfer, but white adipose tissue does not. One has to be careful because it's not this transfer of mitochondrial material all the time between many cell types, but this is not what we're showing here. We're not fragments. It's the transfer of the entire organelle, like you would see in autophagy in autophagic processes. So the answer is yes, we're considering them, and actually we're following this idea right now that this similar process might be taking place in thermogenic mitochondria fat tissue.
Thank you. Esther.
Yeah, beautiful story, Anders. Very intriguing. I was wondering, one of the hallmarks of an aging heart is the increase of fibrosis. So do you think the increase in fibrosis, is that a cause or a consequence of the decrease in macrophages in your aging heart?
Yeah, that's an interesting question. Well, if you follow Ruslan Mesitov line of thought, you know, in this paper where he sees the studies, the crosstalk between the two cell types in isolated systems, how they regulate each other, which is a way that we think of this just for the sake of because we didn't know about this. So that seems like a very reasonable system. The fibroblast produces macrophage supporting factors. The macrophage produces fibroblast protein factors. If one of them fails, then you run into problems. If the macrophage fails, then eventually the mesenchymal cells becomes self-sufficient to support themselves, and then you run into problems of fibrosis. That's his model, and that's the way we start thinking about this. When we eliminate macrophages for 3 weeks, we see diffuse fibrosis and low grade but diffuse fibrosis. Older animals eventually develop also. We can establish a cause effect other than if we deplete, we see this. We don't know how direct this is. So part of the study that we're doing is shifting now to understand the mesenchymal contribution or composition of the heart, of the myocardium.
Yeah, very interesting. I have another small question. Do you see the same phenomenon in the same extent also in the right ventricle? Like the left ventricle is a more higher pressure system and the right ventricle is low pressure?
Yeah, that's a good question. We're getting this before, but again, because we're so purely immunologists, we were not very worried. We were not very curious about that question, which I guess is very obvious in the cardiac field, you know, to correlate the stress with this. We haven't. Unfortunately, we always looked at the left ventricle and then switched to a different question, but we have not done that, you know, accurately, and that's a great point. So I cannot give an answer.
Thanks.
Thank you. Just Mabruka was first, sorry.
That's really very magnificent talk. I have plenty of questions as well, but I'll summarize my questions to two. So my first questions is, do macrophages, the cardiac macrophages, need to be in physical contact with cardiomyocytes in order for Exophys to be shed and taken by macrophages, or they can be shed away and you can detect them in serum? I mean, I'm thinking about...
Yeah, so the answer is yes, they can be produced without macrophages in contact, and we know this because when we deplete macrophages, in the short term, in the long term, you end up having secondary problems of this autophagy machine rate, so you stop producing more Exophys. But in the short term, the first few days, what we see is an accumulation of Exophys. You know, simplistic system is that the macrophages are not there to remove them, and before other problems appear, you just get an accumulation of Exophys. So that shows that the system is not initiated by macrophages, which was one thing that we initially thought and was obsessing one of the reviewers, and that was very irritating because in the end we say, who cares? But you know, we have these. I mean, not who cares, we care, but it's not important for the point that we're trying to make. Mechanistically, it might be interesting, but we saw that if you eliminate them, you get more in the short term. So the system is independent of the macrophage. And I'm sorry, what was the second?
I was thinking about patient with heart failure. Can this be used as a prognostic value? So can we measure
Sure.
serum?
We just looked whether we could see some of these exophys in healthy mice. We didn't look in stress animals, and we couldn't find any. I have to say that the search was not very exhaustive, but in the obvious simple experiment, we could not find by flow particles coming from first in our heart fluorescent heart mouse model. We couldn't find any. That was obvious. Of course, it's interesting to think, if you induce stress, whether they would start appearing elsewhere. I would argue that the phagocytic system has to fail very much for these particles to escape. The system seems to be quite effective, but hey, it's hard to tell.
So one last question: are you expect when you reintroduce macrophage back to cure the phenotype? The heart will come back and function normal?
Yeah, that was one experiment that we did specifically, and yes, so we did, because the C169 DTR system is reversible. If you stop giving the diphtheria, then the population comes back. In our hands, in around a week, the macrophages are back to normal or even a bit higher. We did an experiment where we depleted, got into dysfunction, and then allowed the macrophages to go back again. As we stopped giving the toxin, and then the system recovered functioning very well.
Great, thank you.
Jesse?
So my question actually is very similar to Mabruka's question in terms of how the exopher accumulation can influence the inflammatory response during injury. First, I wanted to know whether you thought it influenced hematopoiesis toward certain lineages that could replace maybe the resident macrophages. But then secondly, when you get other phagocytes, like a neutrophil coming into a site of injury, are they now taking up the exophers?
That's a good point. We haven't looked either of those points, so it's just speculative at this point. So it's a good point. Whether these could be some sort of alarm signals that reach the bone marrow we don't know. We actually haven't. I don't know how we would. I guess we could purify and give from one mouse to another and see if that elicits anything. And that's a good point. And with other phagocytic cells would take over after inflammation, and that's a good point, because when we do myocardial infarction, there's an experiment in which we did myocardial infarction in control versus depleted, and without macrophages, everything, the animal dies very quickly, so the heart is already probably in a bad place. But if you have macrophages, it doesn't matter. That's in the literature there. The resident population shrinks down, and the inflammatory population, which for all purposes is a different thing, appears. The problem there is that there's so much destroyed myocardium that you cannot study the system like we did before. Basically, it's just dead cells invaded by a new type of. So it's more that the environment that they find is different. So the cardiomyocytes are not in that particular area, are not active anymore, and that's where you see accumulation of inflammatory cells. Arguably, it could happen. I think the neutrophils are not so keen in using MERTK for uptake of material. Not that it has to be the only mechanism, and also the particles are quite big compared also to neutrophils, so they're more or less similar in size, anything from 10 to 10 microns in normal conditions. Whether this changes with stress is something that we haven't looked in detail. Like, for example, after an infarction, or if you use a cardiac stress in any way, or hypertrophy, or any of these systems. So that's all out there, reaching to explore, and we haven't, and we're not doing anything about that.
Marit?
So hi, Andres. It was a really great talk.
Hi,
Thank you. Hi. So I had a question about your aging model, because basically there you say you have less macrophages in the heart, right? And I mean, during aging, you also have the myeloid bias. Just wondering how you were fitting that together.
Yeah, well, having more myeloid output, or even in some organs to have, doesn't mean that you get the bona fide resident macrophages to increase to the same extent, or even alter the phenotype, which is something else that can happen. In the heart, in the particular case of the heart, we have not much actually, and maybe we should have done with other organs. But in the heart, the decline is very strong, even if the myeloid bias appears. I would say a little bit later, by 1 year, still start seeing a switch, at least in our hands. But the decline that you saw is very strong. It's
And do you have any reason for the decline? Do you have any idea why that goes so rapidly?
I wish, because I think one hypothesis in mind is that this might be key for organ function decline with age.
Yeah.
Regardless, it's intuitive that if you lose this or other population of resident macrophages, it's not going to be good and nice for the organ. They're there for a reason. And the particular case of the heart, that's what we're trying to see. What is the cause, or at least could we revert this in any way? So that's what we're looking into. It doesn't make sense. Getting old doesn't make sense in general, but perhaps the loss, the very 100% of people will have some sort of heart failure at one point or another if they get older. This is true as life, and why is this, and is this even reversible at all? And that's what we're looking into.
Very interesting. Thank you. Yeah.
Thank you.
Okay, I guess I have the last question. So thank you so much for joining us, Andres. My question will be about the mitochondria that remains. So usually if you remove mitochondria, almost there is a specific rheostat which allows them to reestablish the original number of mitochondria. So what happens to the mitobiogenesis program if there is less or more macrophages when these processes are working or not?
Yeah, we tried to look into that. There were some questions from the reviewers also that we had at the beginning, and we couldn't. I wasn't very satisfied with the technical approach that we did. So the answer is not very convincing. We got some evidence by looking at PGCA. What is it called? PGC1A, I think, alpha levels, and another surrogate. Don't remember now. Of mitochondrial biogenesis, that it seems to be increased. Like there would be more of mitogenesis when you depleted macrophages, which I guess sort of made sense if they were dysfunctional. I don't know how this mitochondrial rheostat works, but you would argue that if it's dysfunctional, if it's based on metabolic output, then you would call for more production to compensate, and that's why we get more mitochondria in the first place when we deplete macrophages. It could be also that they cannot live freely, but presumably also because they're produced at a higher rate. So the mild, maybe not very convincing evidence is that the mitogenic process is triggered. But again, I wouldn't put my hand in the fire on that. But that's something that we look not very, again, not very. We're immunologists. That's the only excuse that I have. I find a lot of these important and cool questions.
Yeah. So if the macrophages could drive the mitobiogenic process, that would really argue for rejuvenation of the cardiac muscle.
Yeah. I would argue that this is an indirect effect through controlling this process that they might influence. Whether they have a direct influence on mitogenesis could be, but I don't even know if it would be related to this mechanism, or at least indirectly.
And a final question is, what's the role of thyroid hormone in all this? Can
I ask why you're asking this? We have not explored this. That's sensitive.
Yeah, because I believe thyroid hormone is very important for the cardiac rejuvenation or health of the muscle and the mitochondria. So I was wondering if thyroid hormone also drives these processes between the macrophages and
the only thing I know about this is it was important for the metabolic switches early on. I think the thyroid signaling, but I have no idea. It's something that I have to admit that this is something that we haven't even considered. So that's again our bias. But it would be interesting to see if there's. And I'm just trying to see. I'll think if there's any obvious links. There might be in the literature between thyroid hormones and macrophage. Same thing, macrophage maturation. What we're trying to establish, independent of this, is at least in early life, when the two, both the immune system and the cardiomyocytes are kicking up and changing very much their properties, as I showed before with the work of Maria, is what drives what. And we've been in this for a while, and we're not convinced. They match in time. So in our heads, is that there's something that very dramatically influences both of them, or is that one is talking to the other, or the other one is talking to the other, and this is what is this dependency between the two systems. Bottom line, in the end, in the adult, you have to have a two functional system: the macrophage pool supporting, and the cardiomyocyte that is become fully functional and fully oxidative, and so on and so forth. How that comes about, it's I don't know.
Yeah. Well, thank you for answering my questions.
I tried. That was a tough one.
Well, it was good seeing everybody. Have a good weekend, and thank you, Andres, for joining.
A pleasure.
See you all next week.
Thank you.
Bye bye.